What exactly is the gut microbiota? Is it the community of bacteria, viruses, and fungi living in our gastrointestinal tract? Together, they are genes from what we call the microbiome. So when we talk about the microbiome, we are not just referring to the organism themselves, but to the entire collection of microbial genes. Next up, what does a healthy gut microbiome look like? It's all about diversity and balance. Primarily, the gut bacteria come from two phyla, Bacteroides and Firmicutes, and within this, they help regulate the immune system. Did you know your microbiome start forming at birth? For a long time, scientists thought babies were born sterile, but now we know. Microbial exposure may begin even in the womb. The real actions start at birth. It's influenced by the mode of delivery, breastfeeding, antibiotic, and surrounding. At first, the infant gut is home to facultative anaerobe, but gradually shift to anaerobe. By age 2, the microbiota resembles of an adult, though it still changes over time.
Next is crosstalk between the gut and the immune system. In the gut, immune defense relies on gut-associated lymphoid tissue, which include mesenteric lymph node, peripatches, and lamina propria immune cells. The first line defense is epithelial cell, which equipped with pattern recognition receptors such as toll-like receptor and node-like receptor. Those receptors detect microbial-associated molecular patterns, which help differentiate between commensal, microbes, and pathogen. This ensures that immune system does not overreact to the normal gut flora. When potential threat is identified, immune cell beneath the epithelial layer, mainly dendritic cell and macrophages, are respond. Pattern recognition receptors are expressed on both dendritic cell and macrophages to help differentiate between commensal, microbes, and pathogen. For dendritic cell, it samples microbe directly through transepithelial dendrites or M cells. It also migrates to mesenteric lymph node to present antigen to naive T cell to activate adaptive immunity, which promote regulatory T cell in response to commensals, or helper T1 and helper T17 in response against pathogen. For macrophages, it involves in phagocytosis and microbial killing. They also produce pro-inflammatory cytokines to avoid unnecessary inflammation. It also involves in clearing apoptotic cell and maintaining tissue repair. This cell also activated in gut-associated lymphoid tissue. It produces secretory IgA, which help neutralize pathogen without disrupting beneficial microbe.
IgA is produced by plasma cell in mucosal area, especially in the gut, and secreted as secretory IgA in peripatches. This cells switch from IgM to IgA, guided by dendritic cells and T cells. This is because IgA is better for mucosal protection. The illustration can help us to understand how they work. IgA is secreted in dimerate form by the polymerase immunoglobulin receptor on gut cells and becomes secretory component that protect it from degradation. Secretory IgA acts as the first line of defense, neutralizing pathogen and toxin before they reach the epithelial cells. The illustration above shows how the neutralization of pathogen occurs. Secretory IgA plays an important role in protecting mucosal surface like the gut. It binds to harmful microbes and toxins in the gut lining, neutralizing them and blocking their attachment to the gut wall without triggering inflammation. This helps prevent gut damage and discomfort. Secretory IgA also gently coat beneficial gut bacteria, controlling their growth and location while preventing unnecessary immune response. This helps maintain mucosal immune balance or homeostasis. Finally, secretory IgA traps harmful microbes in the mucous layer, preventing them from crossing the gut lining, a process called microbial translocation. It also strengthens the gut barrier by preserving tight junction between cells and neutralizing harmful substances like lipopolysaccharide. Without enough secretory IgA, the gut becomes exposed to imbalance, dysbiosis, and inflammation, increasing the risk of disease.
Now, I'll be talking about the impact of the gut microbiota on our health and how imbalance in our gut bacteria, also known as dysbiosis, are connected to various diseases. First part is gut microbiota dysbiosis and disease. Dysbiosis weakens the immune system, contributing to autoimmune disease like type 1 diabetes and rheumatoid arthritis. It also can cause allergy, asthma, and inflammatory bowel disease like Crohn's disease and ulcerative colitis. Let's move on to beneficial microbe and pathogenic imbalance. Some bacteria are super helpful, like Akkermansia mucinifila, which protect the gut lining and improve metabolism. Bifidobacterium, also beneficial microbe, which help to fight inflammation and support immunity. However, when harmful bacteria like Clostridium difficile or certain strain of E. coli take over, often after taking antibiotic, they can cause serious infection and inflammation. So, how do we fix it? We can fix it by therapeutic approach. First is by taking prebiotic, which are fiber that feed good bacteria. Next is probiotic, like lactobacillus and bifidobacterium, found in fermented food like yogurt and kimchi. They help to restore balance in our gut, support immunity, and may also help with inflammatory bowel disease and allergy. Lastly is fecal microbiota transplant, which used to restore microbiota balance, especially in severe Clostridium difficile infection.
Gut-brain axis (GBA) refers to a bilateral communication between the gut and the brain involving neural, humoral, immune, and microbial pathway. There are three signaling routes. First is the neuron immune pathway, where the gut information release cytokines and can influence the brain function and mood. Second is neuroendocrine pathway, which includes the HPA axis, where stress triggers the release of cortisol and gut hormones such as serotonin, ghrelin, which impacts the brain signaling. Third is the microbial signaling. Our gut microbiota produces short-chain fatty acid like butyrate and communicate with the brain via the vagus nerve. This axis plays a major role in mental health. An imbalance in the gut microbiota, called diabolosis, is linked to anxiety, depression, and also autism spectrum disorder. Emerging research even suggests that probiotics, or also known as psychobiotics, might help regulate mood and behavior.